Hello! We are starting B4, and we are going to be looking at gene interactions. We're going to be looking at exceptions to Mendelian genetics. We are going to find examples where the law of independent assortment is not followed. Remember, the law of independent assortment is that genes for different traits are going to be inherited independently from each other. We're also going to see that there's not necessarily always a dominant and a recessive allele. There's not always a dominant allele that's going to completely mask a recessive allele. So we're going to be looking at those examples, non-Mendelian examples, and we're going to be starting out with something that's not in your notes, something called linked genes. So linked genes. Genes that are close together on a chromosome tend to be inherited together. Right? They're not going to show independent assortment. And so because of that, you're going to have some gamete types being more common than others. So our first example, showing you exceptions to Mendel's laws. So we're talking about linked genes that are on the same chromosome. So when genes are on the same chromosome, but they're really far apart, like this purple and the blue one, they can assort independently due to crossing over. Right, when the homologous chromosomes, they grab each other, exchange DNA. Because they're far apart from each other, they are able to do that. But these ones that are really close together, right, they're not going to be exchanged. They're going to stay together. They're not going to be exchanged independently. So these alleles that are so close together on the chromosome, they're going to be passed as a unit to the gametes. So this is going to be passed as a unit. They're going to go together. So you're always going to find these two traits together. So you're going to find more of these traits together, more of these gametes with these particular traits than you normally would have. So these genes are linked because they're so close together, they are not able to assort independently when crossing over took place. Our next example that we're going to look at is called incomplete dominance. In this case, we're going to have 3 different phenotypes in the monohybrid. So we're going to have red and white, and then they're going to produce pink. So 3 different phenotypes in the monohybrid, and use only capital letters, and use primes, and I'll show you what I mean by that. There's going to be an additive effect. The genes are going to add up. And the offspring are going to show characteristics in between the 2 extreme parental characteristics. This is kind of like the blending idea of genetics. Red one and a white one, and you get pink. So here is an example of incomplete dominance. Notice how we write it. So we have 2 parents. Notice we have red. And look how you're going to get red. You write a capital letter. R1, R1. This is red. Let's do it this way. So red. We're going to have R1, R1. Then I'm going to put the heterozygote in between. Pink. So it's something in between both parents. This is the heterozygote. Notice how I'm going to write it. R1, R2. And the white is going to be R2, R2. So we have red and white, and one is incompletely dominant over the other. Let's say we had red and blue were the two extremes, and then the one in between would be purple. So let's go ahead and do a problem with this one. Let us cross a heterozygous. Let's do a heterozygous cross. R1, R2 times R1, R2. Okay. So let's do a heterozygous cross. Let's set up a Punnett square here. Just broke a line there. So setting up a Punnett square. R1, R2. These are both pink. Alright, so they're both pink flowers. So those are the parents. Look at the F1s. R1, 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 R2, R1, R2, R2, R, R2. You see that? Now what we're going to do is we're going to find the genotypic ratios and the phenotypic ratios. So let's find the genotypic ratios first. I'm just going to put a G like that, genotypic ratios. So we have 1, R1, R1. We have 2, right here. R1, R2, and we have 1, R2, R2. Now let's look at the phenotypic ratios. Phenotypic ratios, we have 1 red, we have 1 pink, and put the 1 there, one of those, and we have 1 white. So notice, the genotypic ratio is 1. I'll put a 2 there. You can see it. Oh. 1 to 2 to 1. And so here's the genotypic ratio, and the phenotypic ratio is 1 to 2 to 1. So look at this. Genotypic ratios and phenotypic ratios are the same. 1 to 2 to 1. Red, 2 pinks, 1 white. Let's compare that now to the complete dominance. An example with complete dominance. So that was incomplete dominance. And we're going to see how that is different to the Mendelian genetics. And we're going to call that complete dominance. I'll just put DON. So in this case, we're going to have only two phenotypes. This is going to be red. And there's two ways to get the dominant phenotype. Homozygous dominant and heterozygous. Little r, white. Only one genotype to get that. So there we have some differences already. Not only the way we're writing it, but we had red, pink, and white. So incomplete, we had 3 different phenotypes. Here we only have 2. And here we have 2 ways. Look at the heterozygote. Oops, this would be green. So look at the difference of the heterozygote. The heterozygote shows the dominant trait. Over here, the heterozygote shows a mixture of the 2 extremes, the red and the white. So it will be pink. So let's do continue on with our comparison, and let's do a heterozygous cross. So let's do a Punnett square. Let me do it. So we can compare them next to each other. So we're going to do a heterozygous cross. Get my orange here. Try to make them across from each other so we can see them as a comparison. So big R, little r. Big R, little r. Fill it out. And then let's come up with our phenotypic ratios and our genotypic ratios. In this case, our genotypic ratios, let's put that line here. Our genotypic ratios, so put them here so we can see them across from each other once I move this over. Genotypic ratios, we're going to have 1 homozygous dominant, 2 heterozygous, 1 homozygous recessive. The purple. Phenotypic ratios, and make sure you know the differences between these. These 3 are going to be the same. So you're going to have 3 red, 1 white, no pink. So in the incomplete dominance over here, take a look at that. We have a different way to write it, capital letters, numbering, or you can write it with a prime. That's one difference. Now the difference is this is going to have 3 different phenotypes. Let's see if I wrote it. Oh. Alright, so let's start over. I moved it over here so you can see it. Over here we have 3 different phenotypes. Alright, so one difference was the way they're written. Another difference is incomplete dominance has 3 different phenotypes. Complete dominance only has 2. Incomplete, red, pink, and white. Complete, red and white. With the complete dominance, the dominant recessive, the dominant allele was able to mask the recessive. But over here in the incomplete, there is no masking. Let's start with the difference of the heterozygote. In this case, because the dominant allele masks the recessive, the heterozygote has the dominant trait, same as homozygous dominant. But over here, because there's no masking, and that's why it's called incomplete dominance, they're not completely dominant over another one, we get a mixture. So the heterozygote is a mixture. So in red and white, we get pink. In red and blue, we get purple. And then we did a heterozygote cross. So right here, we did the heterozygote cross. And we came up with genotypic ratios and phenotypic ratios. With the incomplete dominance, our genotypic ratios and phenotypic ratios were the same. 1 to 2 to 1. Genotypic ratio, 1 R1 R1, 2 R1 R2s, 1 R2 R2. 1 red, 2 pink, 1 white. But over here with the complete dominance, notice the genotypic ratios and the phenotypic ratios are different. The genotypic ratio is 1 to 2 to 1, which is the same as the incomplete dominant, but here's the difference. The phenotypic ratio is different because the heterozygote is different. There's masking. So all those with one of the dominant alleles are going to be red. So their phenotypic ratio is going to be 3 to 1 in the heterozygote cross. Sometimes they do not write the incomplete dominance with the 1s and the 2s. Sometimes they write them in the same way as they do the complete dominance. And we will do some problems like that, but you should be able to figure out that it's incomplete dominance either way. So let's look at this. Complete dominance. The dominant allele completely masks the recessive. We only have 2 phenotypes. When you mix a red and a white, you're still going to get red. Incomplete dominance. No masking. Neither allele is dominant. You cross a red and a white, and all the offspring are going to be pink. All of them. So we cross a red, R1, R1. That's a 1. With, so red and a white. This can only give R1s. This one can only give R2s. So all the offspring are going to be pink. So this is red. This is white. These are all going to be pink. So we had incomplete dominance, where neither allele was dominant, but they combined. And they displayed a new trait by mixing the 2 alleles together. But now we have another kind that is called complete dominance. In this case, both alleles are fully dominant. Alright? Fully dominant. They're equally and fully expressed. So both of the alleles of the genes are dominant. And the heterozygous phenotype is actually going to express both of the traits. So let's look at this. So both the heterozygous alleles are going to be fully expressed simultaneously in the phenotype. And both of them are going to be equally expressed. Alright, so it's not like the incomplete dominant, where there's a mixing in the heterozygote. So look at these examples. The examples we're going to be talking about later on is blood type A and B and sickle cell anemia. So keep those in mind. So blood type, just the A and B, they are codominant, and sickle cell anemia. And just looking at the pictures, which one you think is which? Remember, codominance, we're talking about the heterozygotes here. If we have a red and a white, alright, red and a white, incomplete, remember there's going to be like a mixing of the two phenotypes. And codominance, equal dominance. And if you guessed that, you were correct. Notice that one has red and white. But this was a mixture between the red and white, and we got pink. Or here we have red and white. Here we have the red, the white, and the pink. So equal dominance, codominance, both equally present. Incomplete, there's a mixing. Here's another example. Here we have this brown cow and this white one. And look, you have both equally expressed. They are both dominant. And those brown spots all over it. So look, capital letters, both capital letters. Equally expressed simultaneously. Both are dominant. Here's another example with these chickens. Black and white. Alright. Black and white, notice they're all capital letter, they're all dominant. And they're all going to produce, the heterozygote is going to be speckled. Black and white. So black and white. There's no mixing. The heterozygous phenotype, has both traits equally expressed. Another non-Mendelian example is pleiotropy. One gene will have many effects. So an allele can affect more than one phenotype characteristic. It may control several different and unrelated traits. Here's an example. One gene will have many effects. You might not think they were related at all. One example is Marfan syndrome. Tend to be tall, thin, with long arms, legs, and fingers, nearsighted, weak aorta walls, and all because of this. All these characteristics are due to an inability to produce a normal extracellular matrix protein called fibrillin. Extra means outside, which strengthens elastic fibers and allows extracellular components to adhere to one another. So one problem, one allele affected all those things. Another example of a pleiotropic gene, piebald. Irregular patches of two colors. It can affect your fur coloring and hearing in some dog breeds. Sickle cell. We're going to talk more about sickle cell. That's an example of pleiotropy, one gene having many effects. Sickle cell anemia is just due to one mutation. You have one wrong amino acid. So this is what the normal blood cells look like, nice and round. Look over here. That's why they call it sickle shape. Look what happens. It looks like a sickle. Because that change of the amino acid changed it from hydrophilic to hydrophobic. And we'll see that has many, many effects. So pleiotropy. Look at those sickle cells. So 2 copies of the sickle cell allele. All hemoglobin is the sickle cell variety, so all of their blood cells. Abnormal hemoglobin crystallizes when oxygen content of blood is low, causing red blood cells to become sickle-shaped. And look what happens. It causes breakdown of red blood cells, clumping of cells, clogging of small blood vessels, accumulation of sickle cells in the spleen, physical weakness. Look at all these things. Physical weakness, anemia, heart failure, pain, fever, brain damage, damage to other organs, spleen damage. Why? Because that one allele. And some other things. Impaired mental function, paralysis, pneumonia, other infections, rheumatism, kidney failure. So pleiotropy. One gene, many effects. Still looking at sickle cell anemia, because remember we said that was an example of pleiotropy and codominance. So pleiotropy, one gene influences several traits. An example, we talked about sickle cell anemia, and we looked at all the different symptoms that it results in. That is, if it's homozygous, all those different traits we looked at. And let's look at why. So the sickle cell allele, looking right here, make red blood cells produce abnormal hemoglobin molecules. These molecules tend to link together and crystallize, especially when the oxygen content of the blood is lower than usual because of high altitude, overexertion, or respiratory ailments. As the hemoglobin crystallizes, the normally disc-shaped red blood cells deform to a sickle shape with jagged edges. Sickle cells are destroyed rapidly by the body, and the destruction of these cells may seriously lower the individual's red blood cell count, causing anemia and general weakening of the body. Also, because of their irregular shape, sickled cells do not flow smoothly in the blood and tend to accumulate and clog tiny blood vessels. Blood flow to body parts is reduced, resulting in a lot of those symptoms: periodic fever, severe pain, and damage to various organs, including the heart, brain, and kidneys. Sickle cells accumulate in the spleen, damaging it. Blood transfusions and certain drugs may relieve some of the symptoms, but right now there is no cure. Okay, let's look at the codominant part now. Over here it says, in most cases, only people who are homozygous for the sickle cell allele suffer from the disease. Heterozygotes who have sickle cell allele, one sickle cell allele, and one non-sickle allele are usually healthy, although in rare cases they may experience some pleiotropic effects when oxygen in the blood is severely reduced, such as very high altitudes. These effects may occur because the non-sickle and sickle cell alleles are codominant. Both alleles are expressed in heterozygous individuals, and the red blood cells contain both normal and abnormal hemoglobin. Heterozygotes are said to have sickle cell trait. Another non-Mendelian example is epistasis. And if you remember, that's the word that I told you to write on the second page, the dihybrid page of our genetic problems. So epistasis is a form of gene interaction when one gene locus masks or modifies the phenotype of a second gene locus. There's different types. So for example, when a recessive pair of alleles at one locus prevents the expression of a dominant allele at another locus, a covering up has occurred. And the clue that you're dealing with epistasis is when you do a dihybrid heterozygous cross, you do not get the expected nine to 3 to 3 to 1 phenotypic ratio. You don't get it. Get something else. Here's an example of epistasis. The coat color in rodents. You get a 9 to 3 to 4 ratio instead of 9 to 3 to 3 to 1 ratio. Because CC, 2 little c's, homozygous recessive, is always expressed no matter what the genotype is at the A locus. So the epistasis of CC, homozygous recessive, over the A, over the dominant allele. So if you look at this example, instead of C's we're going to be using A, so let's look at 2 little A's. So 2 little A's, 2 little A's, 2 little A's, homozygous recessive. Oh, that doesn't have a little b. So these 2 homozygous recessive A's are going to mask the second set, as if it's not even there. So basically, it's kind of sort of act like a dominant, and it's the homozygous recessive A's mask that one. So it's not even, as if it's not even there. So that changes the 9 to 3 to 3 to 1 to a 9 to 3 to 4. So you clue that this is epistasis. Both parents are heterozygote. You would expect a 9 to 3 to 3 to 1 ratio if we have complete dominance, but instead we get a 9 to 3 to 4. Notice in both cases they equal 16. Another type of non-Mendelian genetics is multiple alleles. So sometimes there are more than 2 alleles that are govern the phenotype of a particular trait. And our example is going to be blood type. So in blood type, we're going to see the presence of more than 2 alleles for this particular trait. So blood type is going to be governed by 3 different alleles. Let me actually draw it out for you. So the human blood type is actually going to be governed by 3 different alleles. We have A, and we have B, and we have O. The human blood type is governed by 3 different alleles. But each person only gets 2. So each person only has 2 of these 3 alleles. So there's 3 possibilities, but you only get 2. So you're only going to have 2 of these 3 alleles in your DNA. O is recessive. So to get A blood, you're going to have AA or AO. B blood, be B or BO. O blood, O. If we recall, when we talk about codominance, we said that A and B are codominant. So that's another type. They're equally expressed. So for blood type, A and B are going to be codominant. Both A and B are going to be equally expressed. Alright, so it's codominant. Also, we have the complete dominance as well. So in this case, we said that O is recessive. Right, O is recessive. So when we have AO or BO, the B and the A are completely dominant over the O. So blood type shows codominance with the A and B, and it shows complete dominance with the more the Mendelian type. So complete dominance with O being recessive, and AO is the same type of blood as AA. They look the same. The A is masking an O. Both of these are going to look the same. I'm not saying they're necessarily equal, but they're going to look the same. So A and B alleles are both dominant over the O. The O allele is recessive. Codominant, A and B are equally expressed. So blood type in humans inherited by multiple alleles. So we have 3 different alleles and 4 possible phenotypes: A blood, B blood, AB blood, and O blood. So let's look at here. Blood types are dependent on whether an antigen is present and which one. The plasma, so here the plasma has antibodies to the antigens that are not present on their red blood cells. An antigen basically is anything that can cause responses from antibodies. Antibodies are going to react to the antigens, going to fuse with them and destroy them. Red blood cells have a particular glycoprotein on their surface which acts as an antigen. Okay, that's important right there. So, if you're given the wrong blood transfusion, mixed blood types, this could lead to death. Let's go ahead and look at that. So looking at the A, B, and O blood groups. Antigen. So A blood, so looking at A blood, the reason why it's called A blood is because of the type of antigen it has on its red blood cell. A blood because it has the A antigen. Now, that's on the red blood cell. But now we're going to look and see what's in the plasma. For those people who have type A blood, they have A antigen on the red blood cell, And in their plasma, look what they have. Let me get a different color here. They have anti-B antibodies. So that means if they get any blood that has a B of any type, like B, BB, BO, or even AB, these antibodies are going to seek out that blood, and it's going to coagulate the blood, and the person could die. So what kind of blood can they have? They can have A or O. Alright, let's look at if you have B blood. Why is it called B blood? Because they have a B antigen, or these little purple spiky things. B antigens on the red blood cell. And look at the plasma. They have anti-A antibodies. That means if they get any blood that has any form of the A, AA, AO, or even a B blood, those antigens, those anti-A antigens, are going to seek out that blood, and the blood's going to coagulate, and the person could die. So what kind of blood can they have? Anything that doesn't have an A. Like over here, anything, so nothing, no B's. Sell it. And let me get you a different color here. So over here, nothing with B. Over here, nothing with an A. Okay, let's look at AB blood. Get another color here. Let's see. That color. AB blood. Alright, so this is A blood. This is A blood. This is B blood. And this is AB blood. Why is it called AB blood? Because they have antigens A and B. A and B. And look at this. No antibodies, which is a pretty good thing because they would kill themselves. Alright, so AB blood have no antibodies in their plasma. So, because of that, they can have any type of blood. And that's why they're called the universal recipient. Why? Because of this. No antibodies in their plasma. Alright, next we'll look at O blood. O blood actually is the most common blood. So O blood. Why is it called O blood? Because it does not have any antigens. So no antigens. But look what they have. Both antibodies. So they have anti-A and they have anti-B. So these cannot have A or B. No A and no B. That does not look very good. So no A blood and no B blood. They can only have O blood. Why? If they had A blood, look, they have A antigens here and they have B antigens. The purple ones are the A's, the blue ones are the B's. So if they got A blood, anti-A would seek it out, coagulate the blood. If they got B blood, the antibodies against the B would go and seek out that blood, and blood would coagulate and they could die. But look what they're called. Doesn't make any sense, does it? They're considered to be the universal donors. And why is that? That's because up here, the red blood cells don't have any antigens. So if they don't have any A antigens, B's can get it. If they don't have any B antigens, type A can get it. So, and A and B can get it as well. I actually found that blood type actually is a lot more complicated than this. So they don't usually say universally now that AB is universal recipient and O is universal donor, because they've learned a lot about blood type. And sometimes by thinking they got it right, and bad things happened. Positive and negatives are something totally separate. So positive and negatives are totally separate from the A's and the B's and the O's. So next time, we will talk about the positive and negatives and do some genetic problems and finish up B4. Until next time, be well.